In this video, I'll show you how to use my findings in IPA. I'll show you an example of using my findings in an analysis. I'll show you how to add proprietary findings as a batch using a spreadsheet, or how to add proprietary findings one at a time interactively inside IPA. For this example, we're going to use siRNA screening data that we obtained from GenomeRNAi.org. The data is from a paper where the authors knocked down genes and examined focal adhesion formation with a microscope. These findings can be modeled as a particular gene having an increasing or decreasing effect on focal adhesion formation. Then when you're analyzing gene expression data, the up or down regulation of the corresponding genes can be predicted to have an effect on focal adhesion. For this breast cancer expression data set, those findings are used to predict that focal adhesion formation is decreasing based on the expression of the relevant genes in the malignant cells. To examine this result in more detail, you can display it as a network where you can see the proprietary function and click on a relationship to see the corresponding findings. You can see that this is a proprietary finding and click through to the paper that was cited when we uploaded the finding. Now I'll show you how these findings were created. Here is the simple spreadsheet that was used to format the findings. Some of these columns are required and some are optional, and some of the fields use a controlled vocabulary. We will get into those details later. Note that by including the PubMed ID in the appropriate column, IPA was able to retrieve the reference for you. Next, I'll show you how to upload the sheet into a private My Findings repository, and later I'll show you how you can use a downloadable template to simplify formatting the spreadsheet in the first place. This is QIAGEN's demonstration space for My Findings. Yours will be specific to your institution. I am now uploading that spreadsheet, and all the values in the columns are being automatically parsed and mapped. Unrecognized terms are displayed in red. I have already uploaded these particular findings and created a proprietary function called focal adhesion assay, so that term was recognized this time. You would create a custom node as shown had it not been recognized. In this case, there are a few gene names that were not automatically mapped. Click to the Unmapped tab to show any row where there are unmapped terms. Now you can interactively map those as I am doing here. Once they are mapped, they disappear from the Unmapped tab. You can save unmapped findings for later by exporting them as an Excel file, or you can just discard them. Now I'm saving the findings to the proprietary repository. Now let me take a minute to show you how to use the provided template to create findings. Click on this link to download a pre-formatted Excel template. When you open the file in Excel, all the possible columns are shown. The ones with a red font are required fields, while the ones with black font are optional. The fields that use controlled vocabulary can be populated using the menus that pop up. Otherwise, you can just type in whatever you wish in the other fields. The other tabs in the Excel file provide example findings, hints, definitions, or list the controlled vocabulary terms that are required. Once you've filled in the template, you can save it and directly upload it using the web-based import tool I showed earlier. Note that the findings may take several hours before they are available in IPA, as they are imported automatically once per day. In the final segment, I will show you how you can create a finding directly on a pathway or network in IPA. Here I've created a blank My Pathway by selecting New My Pathway from the File menu. Click on the Build button and select Add My Findings. Now you can add either an existing IPA node to the pathway or create a new node. Here I will select the Node tool and search for GATA4. I select that gene and add it to the pathway. Now I'm going to create a new drug in IPA called X1000. I designate it as a chemical drug and save it. Now I can connect the two nodes to create a finding. To do this, click the Relationship button and drag a line to connect the two nodes. Choose the type of relationship and, if you wish, the species of the gene and the species where the finding occurred. Click the button to create the findings text, which you can edit if you like. Create a reference for the finding, either a PubMed ID or a link and citation to some other source. Enter a project name and save. You don't need to save the pathway. The finding itself is stored in your proprietary database. Thanks for your time. I hope you found this brief tutorial about my findings in IPA helpful.